I want to give you just a little bit of background on Dr. Wright. Dr. Wright is a board-certified neurologist and movement disorder specialist at UCSD's Parkinson's Center of Excellence. He treats people with movement disorders, including Parkinson's disease. In addition, he's an assistant professor in the UCSD Department of Neurosciences, where he trains medical students, residents, and fellows at UC San Diego School of Medicine. He completed his own neurology residency at Columbia University College of Physicians and Surgeons, and then he earned his medical degree from the University of Louisville School of Medicine. When he's not in the clinic or teaching, Dr. Wright conducts research on mechanisms and outcomes of deep brain stimulation, outcomes of continuous intra-intestinal levodopa infusion, gait and balance in neurodegenerative diseases, and clinical research in Parkinson's disease and movement disorders. Today, Dr. Wright is going to share research involving a new technique to regenerate damaged neurons, which reversed symptoms in a mouse Parkinson's model. So Dr. Wright, if you would like to take over, we would love to hear what you have to share with us today.
All right, well, thank you very much for having me. It's great to see everybody. There's definitely a few familiar faces and names in the group today, so happy to see you all and everybody else. We're going to talk about a really exciting technique and experiments that have just been published by a group of basic scientists, cellular biologists at UCSD, and these are really smart people that have done these really well done, rigorously performed experiments, and they have some really surprising results that I think a lot of you have read about, various publications online and in print, and a lot of people are really excited about this, and so we're just going to talk about it. This is a pretty technical article, and so some of these figures are if you actually read the article, it's very complicated and technical and a lot of scientific techniques. But we're going to try to show you some of the figures and really, emphasize the big picture about what was done. And, some of these things are very technical. So if you do have any questions, I'll be happy to answer them. So these are my disclosures whenever I give a talk. These are some of the research support that I get to do clinical trials and others. It doesn't have anything to do with what I'm talking about today. And really, this is not my research today. This is Dr. Fu's research. Okay, so when we talk about, when we talk about Parkinson's disease, as many of us know, there are non-motor symptoms that are very, very bothersome. That can consume a lot of things, but the early features are the motor symptoms. And these are underlined by the primary pathologic event in Parkinson's, which is the loss of the dopamine neurons, whose cell bodies live in the substantia nigra, and they send their axons up to an area of the brain called the striatum. And so, did that advance? Let's see. There we go. Okay, so this is a schematic of that normal circuit. So this is down here is a schematic of all of the dopamine neurons. Here is a dopamine neuron, and it's making dopamine here, and it sends up its dopamine vesicles up to these terminals where they're stored, and then the dopamine is released to the striatum cell bodies that are getting those signals. And so that's represented by this circuit here. Substantia nigra sends up the neurons send up the axons to the striatum where the dopamine receptors are. And when these dopamine neurons have plenty of, they're happy and healthy, and they have making plenty of dopamine, releasing it all the time. You can walk normally, you swing your arms normally. When you do an action, you do it with nice, smooth, and fast motion with no tremor and no stiffness and none of the other features of Parkinson's. And as these dopamine neurons start to get dysfunctional and make less dopamine and eventually disappear, we develop the symptoms of Parkinson's. And those are the ones that are made better when you take usually carbidopa, levodopa, or sometimes a dopamine agonist that restores the level of dopamine to the circuit. And so the person feels better for usually a number of hours, more hours early in the disease. And as many of you know, as the disease goes on, the length of duration of the effect is shorter, and you have to take more and more doses of medication. This is just really a schematic showing that dopamine is involved in other circuits of the brain. Some of the circuits are involved in things like stimulus, reward, pleasure, addiction, and things like that, and also things like hallucinations and things like that. And that's why later on people get problems with hallucinations, because even though this circuit needs the dopamine, this circuit maybe is getting too much dopamine, and you start getting these problems. So the problem in Parkinson's, though, one of the main problems that we're going to talk about today is the death of these cells and the loss of these neurons causing the problems. And that's what this study was studying in mice. Okay, so your brain has a lot of different types of cells, okay? Neurons are like the wires of the brain that are actually making the electricity, and all of the little connections that are going together is basically all of these features that allow you to move and to think and to see and to hear, etc. But they're not the only type of cells in the brain. You also have something called glial cells, which are basically the support cells of the brain. And one of them, most numerous, is called astrocytes. And it's kind of debated about how many neurons and how many astrocytes we have in our brain and glia. It used to be thought that we had 10 times the amount of glial cells as neurons in the brain, but now some people think it's about a 1-to-1 ratio, about 100 billion of each in the brain. But nevertheless, these astrocytes are everywhere in the brain, all around the neurons. Now here's a schematic of what the astrocytes do. So this is a neuron here. This is a blood vessel or capillary in the brain, and these astrocytes are very protective of the neurons. This is a schematic so you can see, but really they're wrapping all around this blood vessel, and basically they are the middleman between the bloodstream and the neurons, because the neurons are very fragile and very important, and we don't want any toxins or any extra high glucose or some kind of other metabolic abnormality to get into the neurons. And so they are kind of like the gateway to the neurons, so they're constantly protecting them and exchanging nutrients with them, and they are everywhere in the brain. This is a schematic so that you can see it, but really in the brain, there are neurons and astrocytes that are packed up against each other really densely and more densely packed than you could imagine. So these astrocytes are everywhere. In addition, after you get an injury in the brain, for example, like a stroke, or even in Parkinson's, these astrocytes will, after the neurons die, the astrocytes will activate and they will fill up the hole in the brain to make a scar of astrocytes, okay? All right. Now we're getting into a little bit more technical stuff, okay? So this is Dr. Fu, who's the senior author of this study, along with some very, Dr. Mobley and Dr. Cleveland are very senior scientists at UCSD. This is the study, and it was published in this journal Nature, which is really thought to be the best scientific journal in the world. And really, it really shows not only was this an amazing discovery and idea, but it's really well done study. And so what they looked at was Dr. Fu has been studying this protein called PTB, polypyridamine tract binding protein, which is a transcription factor. So transcription factors Well, first, before we tell you what a transcription factor is, tell you about how biology works in your cell, okay, in your cells of your body. And so DNA are the building blocks of life, okay? This is a schematic of how it works. And your DNA is the master code, okay? And so it has all these base pairs in these sequences, almost like a computer code. And based on the sequence of the base pairs, it encodes a protein, okay? And the way that it works is you have DNA in your cells that is permanent, it's always there, and then it comes, sometimes some molecules come and they make a copy of a certain gene, and they make that into RNA, okay? RNA is like DNA, but it only has one side. This is double-stranded, this is single-stranded, and it's temporary, okay? It only lasts for a number of hours before it automatically is broken down in the cell. And when you have this RNA, there's other molecules that will turn the code, the message, into a protein. And protein is really the building blocks of life. That's what all of the, the cell walls, the nucleus, all of the organelles, and all of your tissues of your body are really made of proteins, in addition to some other things like carbohydrates and lipids and such. Okay, and so this is how basically the life works. And so all of your cells in your body have all of the genes, okay? However, not all of the genes are activated in all of the time. That's why you have, even though all of the cells have all the same genes, you have muscle cells, you have nerve cells, you have skin cells, and the way that these cells are the way they are is because they have certain genes deactivated that aren't making the message and certain genes that are activated, okay? Now, a transcription factor is one of these proteins that controls which genes are activated in a given cell. And that can determine what kind of cell it is. Is it a neuron, an astrocyte, a skin cell, a blood cell? And so that's how this works. So Dr. Fu discovered this very interesting transcription factor called PTB, okay? And what he found is that this PTB protein is active in certain cell types, but it's inactive in neurons, okay? And when a baby is in the womb, and also probably for a little bit as an early baby and a child, when the nervous system is still developing, these PTB levels in the neurons are very low. And that makes these cells turn into neurons, okay? Now this is a complicated schematic, but basically it shows that this PTB level, when it's bold means that it's high. When this level is high, it inhibits some of these other proteins, these MI proteins that turn on genes that make the cell turn into a neuron, okay? So these are the neurons, these are astrocytes, these are fibroblasts, which are skin cells. And in the skin cells, the PTB is high in the astrocytes, but it's low in neurons, showing that's how it turns into a neuron. So Dr. Fu's group was able to turn fibroblasts, which are skin cells, in a petri dish into neurons by using gene silencing techniques to suppress the expression of the PTB protein. This is another schematic showing how gene silencing works. So this is another diagram. This is actually from the Huntington's literature because there's a lot of work going on with gene silencing.
Now do unmute. Go again.
Oh, sorry. There's a lot of clinical trials going on with Huntington's disease right now in this area. But again, you have the gene, DNA, that makes a master code, temporary transient copy in mRNA that makes proteins. But if there's a way that you can stop this message from turning into a protein, you can change the way the cell works. And there's a couple ways to do this. with these little RNAs. There's something called short hairpin RNAs, there's siRNAs, and there's antisense oligonucleotides. And these are all slightly different because this is the largest molecule, second largest, and this is the smallest. These are the most stable. These are the least stable, the ASOs, but the bigger molecules are harder to deliver into the cells, and they're harder to give as a drug for people. And so that's why a lot of time in the Petri dishes and in the mouse experiments, they use these kind, but in actual people, to make it practical, they're thinking about using these kind. And we'll come back to that later, okay? And the idea was you have the gene that encodes PTB, okay? And you use the gene silencing to make sure that this PTB protein is not made, okay? And so that lowers the level of PTB, and then that turns the cell to go down the path of turning into a neuron, even if it's a mature astrocyte or fibroblast. And so that is, let's see, that is what they did in the Petri dish. They were able to use these short hairpin RNAs to turn fibroblast skin cells into neurons, okay? And that's shown in this schematic here with a variety of very sophisticated techniques using fluorescent proteins tagged to various neuronal markers, okay? And they were able to, so not only were they able to turn skin cells into neurons, they were able to turn astrocytes into neurons. So that's in a Petri dish. That's step one. Step two, what about in an animal? And of course, they do most of their science is done on rats or mice in the lab because they have short lifespan. And so the next thing they did is they injected, using a gene therapy technique, short hairpin RNA into a mouse brain that lowers the level of PTB. And basically, they were able to show that they were able to turn these astrocytes into neurons over 8 weeks. They were transforming them into neurons. And that's really exciting because this is, not like, this is just a small molecule. It's not a big stem cell, which is a very large, and you can't get past the blood-brain barrier. This is a very small molecule turning astrocytes, which are already right in the location that you need, into neurons. Now that was mice that didn't have any, just normal mice that didn't have any issues. Then they used this model in a model, or they used this technique in a model of Parkinson's disease. Okay, and I feel bad for the mice and the rats because they get a lot of horrible things done to them in the lab. But this is kind of how medical research has to be done. This is one of the oldest models of Parkinson's disease. It is a poison called 6-hydroxydopamine, which is a chemical that is very similar to dopamine. And so when it's injected into the brain, it preferentially enters the dopamine neurons. And then the poison goes to work and the dopamine neurons pretty rapidly die. And the model in mice is that within 14 to 21 days, they usually only, first of all, they only inject one side of the brain and they leave the other side intact. And within 14 to 21 days, all of the dopamine neurons die in the mouse, or something like 80% of them. And so this is, obviously that's a little bit different than Parkinson's disease in people, because this is within 3 weeks, it all happens very quickly. And that's not the same as the gradual process that occurs in people late in life. And they usually do this on young mice that are young adult mice, and it rapidly creates these lesions where they're very stiff and slow on the side of the body that is opposite the brain. And then they can do various tests. They're very asymmetrically slow in this model on one side of their body. And so when you give them certain drugs, they will actually circle around kind of run around in circles because one side is weak and the other side is strong. And the medications kind of amp them up, give them a lot of energy, and you can count how many times they circle, and that's thought to be a crude behavioral measure of the Parkinson's phenotype. And then they also do a test where they put the mouse in a little cylinder, and they measure the amount of times that it's touched with the side that's affected by the Parkinson model. And that shows how bad the Parkinson effect is in this mouse. So the more asymmetric, so this is the side that's been affected. If the mouse is only touching it with this front paw on the good side and hardly touching it at all with the bad side, that means that, the model is very strong. Okay, let's see. So anyway, this is a model of Parkinson's that they used this experiment on. So what they did is they injected the toxin into mice, and they had this very severe hemi-Parkinsonian. That means half their body is like Parkinson's phenotype. And then after 30 days, when the toxin had completely done its work, they injected with a gene therapy vector that basically knocked down or decreased the level of that PTB protein. Okay? And then they waited 3 months. And then they did a lot of these tests and compared the before and after. And then they also counted the number of dopamine neurons and the number of dopamine. They used thin layer chromatography to quantify the amount of dopamine in the circuit before and after only after, of course. And they compared it to what we call positive and negative controls, where you had basically almost like a dummy virus that wasn't knocking down any PTB. And so this is just another schematic of the tests. So these are highly technical figures. Essentially what this shows is that you had these behavioral problems that were very asymmetric, where the mouse was circling around and around, that were almost completely reversed by the treatment. Okay.
So everything got better.
So the green is what we call the negative control. That means the mice, they did not put any toxin in them. So they didn't have the Parkinson's phenotype. So there's no asymmetry here in the number of rotations. So the green is the normal. And then after they gave the toxin, they had a group that got the actual empty, which is what we call negative control, and that's the blue, and the orange got the actual thing that lowers the PTB. Okay, so the blue is bad, the orange is the new technique. Okay, and so it shows here. Look at the orange here, how asymmetric it is.
Thanks.
And then this is before, and then 3 months later, you can see how much the orange looks like the green. So it's back to being close to symmetrical. So it's a reversal of this circling around. And here you can see the one that got the empty, it's still the same. Same thing in this, the blue doesn't change, but the orange gets way better, almost like the green. And this is another schematic here. On number of rotations, you can see the orange is the green is the normal, the blue is basically kind of like the placebo but the empty, and then the orange is, you can see after 2 months, 3 months, 5 months, how close it gets to the green, the normal. And so this is pretty cool because this is not only preventing damage, this is a reversal of damage. Which, a lot of people thought that maybe, you can't reverse neuron damage. This is the same thing with the mouse touching that side with its front paw, the side of the cylinder. You can see the green is the normal, the blue is the empty, and the orange after 3 months gets close to the green. So it's kind of a reversal of this abnormal behavior that we see in this mouse model of Parkinson's. This is another thing they did. So unfortunately, what happens a lot with mice is after they do the behavioral test, they will what they call sacrifice and then they take out their brain and they count the number of cells. And this basically shows that here is the good side, here is the bad side, intact and lesioned. And this is what happened so you can see there's all these dopamine neurons on the good side. The bad side, they're all gone after the toxin. And this is with the empty. But when they gave the PTB inhibitor, you can see a good amount of this green is coming back. Now, it's not 100%, but it's a good amount. And this shows what this shows about how much it came back. So this is the intact side, and this is the side that got the empty, and this is the side that got the PTB inhibitor, and it gets to about 30%. About 30% of the dopamine neurons are actually coming back. And these are actually astrocytes that were already there in that scar that were turned into neurons, okay? So that's what it's used the technique to turn astrocytes into dopamine neurons. And there's a lot of these are neuronal markers that they use. They use very sophisticated techniques. In addition, they also checked, that was counting the cells. This is now when they do a chemical test to measure the amount of dopamine in that homogenate, and basically the lesion reduces the dopamine levels to 25% of normal, and the PTB inhibitor restores them to 65%. So not 100%, but 65%. So 30% of the cells coming back and 65% of the dopamine levels coming back. Okay, so that was done using these short hairpin RNAs, which are very powerful, and they use those very often in petri dishes and in mice because they can inject them directly into the brain. But those are harder to deliver to patients, and people are big, have a lot more tissues and thickness of tissues and cell numbers than little mice, and the brains are so much bigger. And so they wanted to try this with the smallest versions technique called ASOs. And so these are much smaller, these are single-stranded, and they can actually be delivered to the brain via just a spinal tap. And so they did a proof of concept in the mice, showing basically that they could use these ASOs to get the same effect. Okay, so it's not quite as powerful, but they were able to get, this is 0 months, 3 months. This is the mouse circling around, and the number of rotations in all of the mice improves, and some much more than others. This is the negative control, and they actually get worse, but this is the orange, the good one. So they were able to show that using the ASO strategy rather than the short hairpin RNA strategy, they could get these same kind of transforming the astrocytes into new neurons and get improvement of the behavioral abnormalities that you see in this mouse model of Parkinson's. Sorry, getting a phone call. All right. Okay, so this is kind of exciting because with these ASOs, antisense oligonucleotides, it's been shown that these can be used to treat real neurological diseases. I don't know if any of you all have heard of Spinraza. There's a very horrible genetic disease called spinal muscular atrophy, where sometimes very little babies, all of their muscles in their body waste away and they can hardly sit up. And then there's a less severe form where you have onset in teenage years and onset in adult. But they found that giving this in a spinal tap, you get a spinal tap and then you get an infusion, they can really affect the survival and the motor outcomes in these patients with SMA just by delivering it into the spinal fluid. And then there's a huge trial, which I'm very involved with right now, on Huntington's disease that are using ASOs to try to reduce the amount of the abnormal protein in Huntington's disease. And that's a big clinical trial that's going on right now, and we'll be finding out the results hopefully in about a year or so. And so the exciting thing about this is if you could, the possibility, rather than having to have a brain surgery where a neurosurgeon has to inject some kind of gene therapy deep inside your brain, maybe you could just give these ASOs in a spinal tap and they could get into the brain that way. Now, the question is, I don't know, the parts of the brain affected in Parkinson's are deep inside, and will a spinal tap be able to give you enough access to those target sites? We don't know yet, but that's a lot of work that has to be done. And so we always talk about the caveats, and this is something all of my patients, when they say, well, how come we haven't licked this Parkinson's yet? How come the research nothing has panned out? Well, one of the big problems is that humans are the only species that gets Parkinson's. And it doesn't occur in nature in any other animals. And the models that we have in these mice, and sometimes they're done in monkeys, they're just approximations of Parkinson's. And you saw with that toxin, the mice get the toxin, and within 3 weeks, they're like all of their dopamine levels is gone, which is not quite the same as what happens in people. Also, those toxin models, they don't get the alpha-synuclein and Lewy bodies that we see in the donated brains of persons with Parkinson's. And so it's not quite the same. So this is why so many things, promising molecules that have looked great in the mice, and then they go to clinical trials, they haven't panned out. The other thing is that this, something to think about is this is really exciting because it has the potential to repair damaged neurons and restore them. But again, we still have to find a way to stop the ongoing damage in the molecular level of Parkinson's, because even if the new neurons are there, they may get affected by the Parkinson pathology. So I think this may be, if someday, if we can also figure out what is the molecular pathological cascade going on in Parkinson's and figure out a way to stop that, and then we could use this technique to make the repairs. And so that's really exciting because, everybody has always thought that, the best that we could do is, we could stop the process, but we're never going to, once the damage is done, it's done. But this new technique from Dr. Fu maybe has the potential of repairing damage. And this is not specific to Parkinson's. You could envision this could be used for strokes, Alzheimer's disease, spinal cord injury is a big one. And so it's potentially a really big discovery if this can be translated from the mouse to people. Now, how does this compare to stem cells? Well, I think this is a little bit practically easier than stem cells because stem cells are big. They're a big cell, and they can't get through the blood-brain barrier and get into the brain. The other thing is that stem cells are unique to the person. They each have their own genetic signature, so you can't put somebody else's stem cells in you because your immune system will automatically recognize it and get rid of it. And so for somebody to get stem cells, they have to donate their cells, and then they have to do a lot of processing in the lab to turn them into neuron stem cells. And that takes months and months and is extremely expensive and individualized to the patient. But this PTB knockdown ASO would work for everybody and is a small molecule that it could easily be given in a spinal tap if it does turn out to work. And it seems like much more fast and practical than stem cells. So that's why I think this is potentially even more exciting than stem cells, but we have to see. Now, everybody's asked me, well, what's the next step? When can I get this? Well, what they're doing next, they've done it in the toxin Parkinson model in mice. Now they're going to do it in some genetic models. That's where you put some of the human alpha-synuclein mutated genes into a mouse brain, and it looks a little bit more like human Parkinson's. And they're going to see if they can undo the damage done in those mouse models with this technique. And if that is promising, then we would presume that they're going to go to the clinical trials. And so clinical trials is a long process. Okay, somebody raised their hand. We're almost finished, so we'll go right to Jerry in a second. So the clinical trials, you start with the preclinical. This is drug development. That's all the work that's being done in the Petri dish and in the mice. Sometimes they do it in monkeys, okay? And then you have phase 1, Phase 1 is just young, healthy volunteers, just to make sure that the new drug is not super dangerous, just to find the safe doses of the drugs in all people, about 20 to 80 people. Then you go to phase 2. That's where you actually go to the people that have the disease that you're studying. And it's about 100 to 300 people. It's powered for safety, okay? It not necessarily have enough people to show that something works, but it does have enough people to statistically show that something is not dangerous and has side effects, and it can give you the signal that something works. And so when you go to phase 2 and it looks like it's safe, and it looks like there's what we call a signal that it may work, then you go to the big phase 3 trials, a lot of people all around the world, that gives you enough statistical power to show that something really, for example, in Parkinson's disease, makes a difference in the evolution of the motor symptoms, non-motor symptoms, and maybe even improvement. And then if that looks good, it goes to the FDA, and it gets FDA approved. And then there's something called phase 4, is when the drug is already approved, they study it after it's approved in many, many more people. And so these are the steps. And of course, you may have seen this with the coronavirus, the COVID-19 vaccine, and they're on hyperdrive right now to try to get that done by, the springtime, early winter, springtime. And so these are the next steps. All right, that's the end of the slides. And so thanks very much for listening. Hopefully it wasn't too technical. And I think we'll take questions now. So the one question is it a permanent reversal of the motor problems in the mice? Well, it seems that way in the mice, but the truth is, I don't know how long they kept the mice, because I think they sacrificed them shortly after they do the behavioral assessment. But they didn't publish that data if they kept some of these mice before they sacrificed them and kept them and let them live in the old age. But that has not been published yet. So we just don't know. Okay, and then it looks like there was another question about glutathione that I can maybe answer to Jody later, because it sounds like that's more specific question. There's a question from Girija. It says, how do you deliver the therapeutic to the astrocytes? Well, it depends on if you're talking about in the Petri dish, in the mouse brain, or someday in the person. And so in the Petri dish and the mouse, they're injecting there's a virus. This is how gene therapy works, where a virus inserts the DNA encoding the short hairpin RNA into the cells directly, and it gets into the cell, and so it's constantly making these blocking RNAs. Now in people you can do that, but that requires a brain surgery, directly a needle going deep into the cells. But if you have the antisense oligonucleotides, the ASOs, they're small enough and diffusible enough that it can be given in a spinal tap. At least in some of these diseases, other diseases, proof of concept in spinal muscular atrophy and Huntington's disease. Well, Huntington's disease, the trial is still going on, so we don't know if it works yet. But in theory, wouldn't require a brain surgery. And so obviously that's more ideal. And the question is, could you even avoid the spinal tap and just give it in an IV? And that's remained to be seen as well. Okay.
I've had DBS. I'm just wondering how this would be affected.
Well, great question. The only honest answer is I don't know. This is so early. This has only been done in the Petri dish and in the mice.
Yeah.
And, it hasn't been tried in people yet. People brains are much bigger, much more complicated than mouse brains. And again, the mouse model that has been tried is not quite the same as Parkinson's. It's an approximation.
Yeah.
And so how would it affect DBS? We don't know. We would just have to see. That's why you have to really do the research and the clinical trials to see how it will work.
I have a question.
Okay.
this point, no issues with regards to dyskinesia due to excessive production of dopamine once the treatment has occurred. Looks like there's only about 30% increase in dopamine producing neurons and 65% increase in dopamine production.
Mm-hmm. Well, what I can tell you, let me share the screen again, and go back to the model. So this is this model where they have two drugs that they give them. One is amphetamine that basically makes, so again, one side of the brain has been lesioned and all the dopamine levels are gone, and the other side is still intact with the dopamine. And so when they give them amphetamine, that makes the intact side really release all of its dopamine, and they turn this way. When they give apomorphine, which is actually a very strong dopamine agonist, this is something, it's not very commonly given to patients, but they can give it as an injection. It helps people, it's a very rapid, powerful dopamine agonist. And what this does is it hits the super sensitive dopamine receptors on the other side, and this actually causes a dyskinesia type phenomenon, and that's why they turn the other way, because they have excessive movements on the affected side. And so this was reversed. Here's the orange going from here closer to the green, and I think this is kind of an approximation of dyskinesia. And so at least as much as this model approximates dyskinesia, it seems to have normalized that. So that's how I would, and again, mice are not people, and what it seems to have, a lot of, over the years, many things have looked great in the mouse, and it hasn't panned out to people yet, but we just have to keep going and keep trying.
Dr. Wright, how long do you think these trials would take before it's available?
Well, it depends. Let's say that, they have another 6 months of experiments on the mouse and it looks good. Then they do the phase 1 trial, which is usually, small amount of people, maybe 3 months, maybe 6 months. Then they do the phase 2 trial, which is probably a year and a half. And then they do the phase 3 trials with large numbers of people. And these take a long time in Parkinson's because Parkinson's is a slowly progressive disease. And so you usually have to follow people for a year and a half, or 2 years to see if there's an effect. Now, maybe if there's true reversal, maybe it can be even shorter. But I think we're looking at 3-4 years minimum before FDA approval. Now, in terms of when the research study might be available to patients in a research capacity, maybe earlier, maybe 2 years, maybe, something like that. A lot of it depends on what happens with Dr. Fu's lab and the results in the future experiments.
Hi,
Who would be administering these cells? Would just a neurologist like you do it, or a specialist?
Yeah, good question. Well, I can tell you it depends on which way you're doing it. In the case of Spinraza, which is the medication for spinal muscular atrophy, just a neurologist does a spinal tap and injects it. And I actually am working on the study for Huntington's disease, and the patient comes every 2 months and gets an injection via spinal tap. And I am the one that actually does it right now. Now, it's thought that maybe if this Huntington's medication becomes FDA approved, getting a spinal tap every 2 months is a little bit difficult for the rest of your life. So there's something called an Ommaya reservoir, which is sometimes created by a neurosurgeon for people that need to get chemotherapy in their spinal fluid space. and that's where they create a little pocket. They cut out a little hole in the skull, and they create a little pocket where you can inject something into there a lot easier than doing a spinal tap. And so that potentially could be injected by any kind of doctor, neurologist. And then if they were going to do the direct gene therapy, where they actually insert the genes into the neurons in the substantia nigra, that would require a neurosurgeon in a neurosurgery where they actually make a hole in the skull and inject the gene therapy directly into the deep parts of the brain. So it just depends on exactly which technique is going to be used.
Do the cells that are affected during the treatment take themselves into the substantia nigra?
Well, I think what it seems like is that you have a scar form when you give the toxin. And so, let's see, starting in the substantia nigra, sending up the terminals to the striatum. If you look at a patient that's donated their brain that had Parkinson's, all these dopamine neurons have disappeared and been replaced by an astrocyte scar. So I think maybe one of the things that's kind of interesting is that these astrocytes are already in the right place. And if you can turn them into neurons, they're kind of already right there. Of course, this has, again, I keep echoing, still has to be replicated in people.
So the dopamine neuron, when it dies, leaves the scar.
Yeah, it dies and it leaves a space, and the astrocytes kind of come in and fill up that space. Form
the scar, and now we're impacting the astrocytes to turn into dopamine-producing neurons.
Right, they're transforming them, reprogramming them from astrocytes to neurons. Yes.
And has there been any research as to how much dopamine a given neuron produces? And whether the astrocyte is functioning in terms of production in a similar manner?
Well, it did actually, in this study, there's a lot of figures with that, because there's a lot of ways that they can measure the action potential, which is the kind of the electrical activity of a neuron, and they can quantify that according to how much dopamine is released. And I believe they did that in the study, and they found that these astrocytes that were converted into neurons really acted very similar to dopamine neurons in terms of releasing dopamine and their electrical activity. And so I think the answer is yes.
So then looking forward, if this treatment approach were approved, it might be, until what's killing off the dopamine producing neurons in the first place is identified and stopped, this might be an ongoing treatment, similar to what
repairing the damage, keep repairing the damage,
filling in the scar holes with new dopamine producing neurons. Okay.
So I think it's just, it's very exciting, because the ability to repair something is really exciting. People thought that, people have been working on stroke and spinal cord injury, which, unlike Parkinson's, we know exactly what causes a stroke. We know exactly what causes spinal cord injury, but they still haven't been able to repair that. And so I think that's why this is so exciting. We might see the ability to actually repair, nervous system tissue.
I have a question.
The astrocytes have, other than just supporting the neurons?
Could you say that again?
These astrocytes, they support the neurons normally. They act as this bridge between the blood vessels and the neurons.
Yes.
Do they have other functions in the brain other than that?
Oh, there's myriad of functions that they do. They kind of there's something called glutamate, which is an excitatory neurotransmitter, and they kind of modulate how much glutamate is in concentrations in various neurons. They help get the toxins out of the brain. There's many things that astrocytes do, and there's actually more than one type of astrocytes. Some that are in white matter, some that are in gray matter. And so, yes, they do. They're very important. And, that is a good question. If these astrocytes have an important function and you're turning a lot of them into neurons, is that going to have consequences? And we don't know yet. But these are all things that we have to think about.
Yeah, I have a question about, does the brain of an early onset person with Parkinson differ from someone who's had it later on in life? And the second question is I know it's maybe an early question, but who would be the ideal candidate for phase 1 if you're already at that, calculating that?
Well, here's the tough problem with knowing what goes on in the brain. We really can only get a good picture when someone passes away. And that almost always, unless there's some kind of car accident or some other coincidental thing, almost all people with Parkinson's in the later stages, their brain looks very similar. And we don't have a way of looking inside somebody's brain at the early stages, whether they're young onset or the more common, onset in 60s, 70s, or 80s. So we don't really know the answer to that question. There are certain, on the whole, patients with young onset Parkinson's seems to have a slower rate of progression than people with the later onset, and it seems like a lot of times they have the classic is very responsive to the medication, but with that response comes some dyskinesias. But we don't know exactly how the brain works and how the brain differs in these. And the truth is that a lot of people think that everything that we're lumping together these days into Parkinson's disease is probably a group of more specific diseases that someday maybe we'll be able to discover all the genes that have been involved, and we'll have 50 different subtypes of Parkinson's that we can more refine them. And maybe there will be some drugs that work better for some types and some drugs that work better for other types, depending on what genes people have. And that's why they're really trying to work to discover all these different genes that are involved. In terms of the phase 1 trial, remember, phase 1 is just healthy volunteers. That's like, young people that are trying to make a buck, I guess, not people with Parkinson's. So those are, healthy volunteers. And it would just depend on, when they usually advertise that in various places. Now, phase 2, who would be the ideal candidate for the phase 2 study? It would be persons with Parkinson's. And a lot of times the tough thing in research, clinical trials, is they like to have everything nice and perfectly controlled. So a lot of times anybody who's had deep brain stimulation surgery, they don't want them in the early research trials because they think that kind of is another variable that kind of confounds their results. And they often in research trials, at least in the early stages, they don't like people where their cognitive function has been impaired. And so they kind of go after these perfect candidates that are people with relatively early Parkinson's that don't have a lot of cognitive impairment. But it depends on the design of the trial, and we would cross that bridge when we come to it.
And in something like this, say you had a person who had cognitive impairment, say this comes to FDA approval, you would not expect that the treatment would change the cognitive function, but rather the Parkinson's motor and non-motor symptoms potentially.
Well, we just don't know. This is. It seems like this method can turn astrocytes into neurons theoretically anywhere. It doesn't, not just in the motor circuit of Parkinson's.
Okay.
So it could just be, it could be in the other regions of the brain too that have been affected. It's hard to know though when you have questions of drug delivery. Presumably, if you give something in a spinal tap, the spinal fluid is going everywhere, and so you would reach everywhere in the brain. But if you're just going to do a technique where you're injecting in high concentration in the motor circuit of Parkinson's, that's going to focus only on the motor circuit. So there's a lot of variables there. But I think that one thing that's very interesting about this is it looks like it can turn astrocytes, into any kind of, Alzheimer's, strokes, spinal cord injuries, and also Parkinson's too. So, I think there's a lot of possibilities. It's pretty exciting, and I think Dr. Fu is really excited about this.
You have another question. What is your comment on the prospect of the increasing dopamine under the pathological condition like Lewy bodies presents? And that sort of tied in with what we were just discussing.
So are you, I guess, the question is, let's say that you have the pathology there and then you give the treatment and the dopamine levels are increased, right? And you could cause maybe too much dopamine to come back. I suppose that's probably the question. And I think that's a very good question. These things have to be tested, and you have to make sure that the right amount is given and it's not overdone, because you're right, there could be a problem where you regenerate too many cells and they're making too much dopamine. And we know that in Parkinson's, sometimes those dopamine receptors get super sensitive, and that you don't want to get to the point where you're having dyskinesias even without taking medicine. And these are all, forks in the road that may happen. So that's a very good question. And I think these are all questions that are going to have to be answered someday.
And that brings our meeting to a close. Many, many thanks again to everyone for their participation today. And again, most especially to Dr. Wright.
Thank you. Thank you, doctor.
Thank you
Thank
for
you
having
so
me.
much.